for joining us for this MDA Engage community webinar on research and treatments for Pompe disease. Let me turn on my video so you guys can see. Oh, here we go. With multiple therapies now in market and more research being done, it is important that you have the information you need to make the best choice on treatments for you or a loved one. My name is Michelle Barrios, and I am the Community Education Specialist at MDA. We are thrilled to have you join us today for this important and educational webinar. The webinar today is part of our larger MDA Engage flagship community series, which focuses on bringing together the neuromuscular disease community around education and social opportunities. Be sure to visit the MDA Engage section on mda.org for updates on upcoming virtual education events. We are recording today's event and will be posting it to the mda.org website for on-demand viewing to ensure that those who were not able to join us live today are able to access this information. Please know that all phone lines have been muted. We will be having a question and answer session towards the end of the webinar. Please be sure to utilize the Q&A window to type in your questions. If you hover over the bottom of your screen, a tray of webinar icons will appear. Click on the Q&A bubble to open the window and enter your question. You do not need to wait until the Q&A session to chat in your questions. As questions come up along the webinar, please feel free to send those in. Before we begin, I would like to say thank you to our speaker, whom you will meet shortly. I would also like to thank our event supporters, Amicus Therapeutics and AskBio. We would not be able to provide community education webinars like this if not for their generous support, so thank you very much. For over 70 years, MDA has led the way in accelerating research, advancing care, and advocating for the support of our families. MDA's mission is to empower people living with muscular dystrophy, ALS, and related neuromuscular diseases to live longer, more independent lives. MDA is the largest non-governmental funder of neuromuscular disease research in the country. Since its inception, MDA has invested more than $5 million in Pompe disease research. Let's review the objectives for today's webinar. Attendees will briefly review Pompe disease, review current treatments for Pompe, learn about developing treatments for Pompe, discuss how to choose the right treatment for you or a loved one. I would like to introduce our speaker for the day. Diana Dimitrova is the Research Project Manager at Oregon Health and Science University. She attended the Bulgarian Academy of Sciences, where she received her PhD in Physiology of Animals and Humans, and received postdoctoral training at Virginia Commonwealth University and Oregon Health and Science University. Diana has completed 60 grants and currently has 10 active grants. She has over 30 publications on many diagnoses, including myasthenia gravis, Pompe, SMA, and many others. And with that, I would like to turn the webinar over to Diana. Diana, can you hear us? Or can you hear me?
Yes. Yes.
Okay, let me pull up your presentation and we can get started.
Okay. Thank you, Michelle, for the introduction. So we can go on the second slide because you already introduced me and my background. So I'd like to briefly go over the Pompe disease history and introducing some terminology that I'll use throughout this presentation. So Pompe disease is named after a Dutch pathologist named Pompe, who was the first to describe the disease in a 7-month-old infant in 1932. His findings were an enlarged heart and muscle weakness, and this baby died. he also, as a pathologist, found accumulation of glycogen in muscle tissue. Next slide, please. Henry Hurst was the next one researching the Pompe disease, and he found out that GAA deficiency inside lysosomes, and GAA is the enzyme that breaks down glycogen, would explain the symptoms of Pompe disease. And I'll have some explanation of this terminology in my next slides. So Dr. Hurst was the one who established that Pompe disease is a lysosomal storage disease, abbreviated as LSD. Other name for this disease is GAA deficiency or type 2 glycogen storage disease. So glycogen storage disease is a group of metabolic disorders characterized by abnormalities involving the use and or storage of glycogen. Besides Pompe disease, there are more than 10 other glycogen storage diseases, but today we'll be talking only about Pompe. Next slide, please. I felt like I need to explain what lysosome is because it's a lysosomal storage disease. So a lysosome is a membrane-bound cell organelle, so that's a particle inside the cell. It contains digestive enzymes, and these enzymes break down complex molecules like glycogen, for which is of particular interest in Pompe disease, into simpler molecules. Glycogen breaks down into to glucose, and glucose is the main energy source for most cells in the body. The lysosomes also break down excess or worn out cell parts. So this process is equivalent to recycling because the products of this breakdown can be reused by the cell for other purposes as needed. And the lysosomes may be used to destroy invading viruses and bacteria. Next slide, please. So the GAA enzyme that I mentioned, the full name of it is acid alpha glucosidase. Other names of it is alpha 1,4-glucosidase or acid maltase. This enzyme helps to break down glycogen in the lysosomes, and when it's deficient, this leads to Pompe disease. Next slide, please. Another term that I used was the GAA gene. So in humans, the GAA gene encodes, or in other words, provides instructions for producing the GAA enzyme. Errors or mutations in this gene cause deficiency in the GAA enzyme, and consequently glycogen accumulates, and that's why Pompe is called glycogen storage disease, and it's type 2. Next slide says more about Pompe disease. So it's an inherited lysosomal storage disorder caused by a deficiency of the enzyme GAA. And when this enzyme is at reduced or absent levels, glycogen accumulates in the lysosomes of muscles and other tissues. And as disease progresses, this glycogen accumulation is believed to lead to the morbidity and mortality associated with Pompe disease, including muscle weakness and respiratory insufficiencies. Next slide just summarizes the cause of Pompe disease, as it happens when the body is not able to break down the complex sugar glycogen for energy. And this glycogen buildup damages muscles and organs. Respiratory and cardiac problems are also issues. And Pompe disease is a genetic disease. And on the next slide, I have listed the 3 types of Pompe disease. The first one is the classic infantile onset. It appears within a few months of birth. And then the next one is non-classic, also infantile onset, that appears at about 1 year of age. We are at OHSU in Portland, Oregon. We are an adult clinic, so we see this third type of late onset, which appears later in the child's life, after 1 year of age, and the symptoms can start in the teenagers or in adulthood. Next slide. Organs affected in Pompe disease are liver, heart, muscles. It expresses the most troubling symptoms are muscle weakness and trouble breathing. And the breathing problem is because, again, of due to muscle weakness of the respiratory muscles. Next slide lists symptoms for the early onset Pompe disease, which show up in infants a few months old to 1 year old. In infants, the symptoms are more intense and progress quickly than in adults. And these babies don't live for more than a year if not treated. And here you can see a list of symptoms in these infants. They have trouble eating, not gaining weight, poor head and neck control. Rolling over and sitting up shows up later than expected or develops later. Breathing problems and lung infections, enlarged and thickening heart or heart defects, enlarged liver and enlarged tongue. And the next slide lists symptoms in late Pompe, late onset Pompe disease. They express in people between 1 year old and 6 year old. They are mostly associated with skeletal muscles, not that much heart. Muscle weakness gets worse over time and will eventually lead to serious breathing problems many years later, especially if not treated. Patients feel weak in their legs, trunk and arms. They have shortness of breath, hard time exercising, lung infections, trouble breathing while sleeping, a big curve in the spine or scoliosis, enlarged liver, enlarged tongue that makes it hard to chew and swallow, and joint stiffness. Next slide, talking about who gets Pompe disease. Since it's a genetic condition. The people who get this disease inherit it from a parent. And it's common that neither parent shows any symptoms. The Pompe disease is rare. In the United States, only 1 in 40,000 people is affected. And it can affect both males and females of all ethnic groups. The next slide shows a diagram of how the Pompe disease is inherited. So it has a couple of parents. Each of them has 2 copies of the GAA gene. So these are the bluish bars representing 2 parts of a chromosome in each parent. And then the affected gene is marked with purple. So in this diagram, each parent has 1 affected gene and the other one is healthy. The type of inheritance for Pompe disease is called autosomal recessive because the person needs both copies of the GAA gene to be affected or mutated in order to be affected by the disease. And the bottom part of the diagram shows the 4 possible combinations for children, because the children get 1 copy of the GAA gene from the father and another one from the mother. So in that purple kit to the left, both copies are damaged. So this child will be affected by Pompe disease. And the girl to the right of the diagram that says unaffected, she inherited 2 healthy copies of the GAA gene. So she will be totally healthy. And the 2 children in the middle are the ones who have only 1 gene affected and 1 healthy, like their parents. So the parents and these 2 children in the middle won't have any symptoms throughout their life, but because they have 1 affected copy of the GAA gene, they are carriers and they can pass it to their children. So in this diagram, the possibility of a child to have a Pompe disease is 25% or 1 in 4. But if the parents have a different set of GAA gene copies, either both copies affected, like the first child, the purple child, or one of the others, then you can draw all possible 4 combinations of genes from mother and father, and the probability of their children to have been to be affected by Pompe will change. Next slide, please. So there are treatments for Pompe disease, and the currently approved treatments are with systemic recombinant human GAA enzyme replacement therapy. So this means we are artificially replacing the missing enzyme or the enzyme with the low level, the GAA enzyme, with another one. but this is a treatment of symptoms, not a cure. And the current standard of care exhibits poor uptake in skeletal muscles, meaning it doesn't enter the target cells very successfully, which is a limitation of its clinical efficacy. This enzyme replacement therapy, abbreviated as ERT, is the currently approved treatment for all Pompe patients. It's given intravenously, meaning through the patient's vein. And the drug is a genetically engineered enzyme that acts like the naturally occurring GAA enzyme, just replacing it. And one example is that Avoglycosidase Alpha. And I'll talk more about it in my next slide. So the next slide lists the current treatments which are developed by a drug company, Genzyme, which is now part of a larger drug company called Sanofi. All of these three drugs, Lumizyme, Myozyme, and Nexviazyme, are enzyme replace therapies. So they replace the missing GAA enzyme to help the body process the glycogen correctly. And all of them are infusion drugs. So Lumizyme is the one most widely used in the United States. Myozyme is for babies and children. And Nexviazyme is for patients 1 year of age and older with late onset Pompe disease. And this last one, Nexviazyme, just got approved last year. Next slide. I kind of wanted to present you briefly the results from the first clinical trial for Myozyme, which led to the first ERT enzyme replacement therapy for Pompe disease in the United States. So about 60 patients were received the investigational drug, and that engineered enzyme was Avoglycosidase Alpha, and about 30 patients received placebo. So placebo is a drug that looks like, smells, tastes, well, not for infusion, but in general, like the real drug, but it doesn't have any active ingredient. And it serves as for comparison to the results from the investigational drug. So the results after 78 weeks in this study were compared to data before these patients started the study drug. Next slide shows this clinical trial data. So the main outcome measure for the trial was a 6-minute walk test. And for patients who were on the investigational drug, Avoglycosidase Alpha, these patients walked 26 meters more than before they started the trial. This is the last row of the table. Well, the patients who were on placebo actually walked 5 meters less. And next slide shows another measure for the effectiveness of the study drug, which is called the forced vital capacity. It's a measure of lung function. So the patients who received the study drug improved their breathing function by roughly 1%, while patients on placebo got worse by 2% after 78 weeks. So during this study, the drug was called Myozyme, and it got approved by FDA. Let's go to the next slide. The component of this drug is Avoglycosidase Alpha. So this is again the engineered enzyme to replace the GAA enzyme. So this drug got approved by FDA in 2006, and the drug was called Myozyme, and it was approved for infantile onset Pompe disease. Later, in 2010, the FDA approved this drug for late onset Pompe disease, and that's ages 8 years and older. And they renamed it for this population as Lumizyme. So this drug Lumizyme, in 2014, the FDA extended their approval to use this drug for kids younger than 8-year-old. And at that time, the Myozyme brand has been got discontinued. So currently in the United States, the Lumizyme is the popular brand of Pompe treatment. Myozyme is still used in Europe, though. And next slide. Lumizyme dose and cost. So the dose is 20 milligrams per kilogram of body weight. It's administered every 2 weeks as an infusion. And the volume infused is determined by the patient's body weight. So it varies, between children and adults, and should be administered over approximately 4 hours. So that's kind of a burden for patients, 4 hours every 2 weeks. And the cost is up to $100,000 for a child for a 1-year treatment, and then $300,000 per year for adult treatment. And the price varies because of the body weight of the people. It's covered by insurance, but again, even if so, you still have to pay deductible and copayments. And next slide is about Nexviazyme. So that's the third drug of all 3 that the Genzyme Sanofi company developed. And I'm going to go briefly over the results of their COMET study that led to the approval of Nexviazyme. So in this study, about 50 people received the new drug, and about 50 people received the currently, or the approved drug at that time, Lumizyme. So Lumizyme contains this Avoglycosidase Alpha enzyme, and the component of the new drug was called Avoglycosidase Alpha. But both of these engineered enzymes have similar properties. The new drug, Avoglycosidase Alpha, is a modification of the previous one, Avoglycosidase Alpha, just a little addition to the molecule. The idea was to make it better received in the target cells. The dose for the two drugs was the same, 20 milligrams per body weight, infused every 2 weeks over 4 hours of infusion. And this part of 49 weeks double-blind study, meaning neither the investigators nor the patients knew whether they were receiving the approved treatment or the new drug, was evaluated at 49 weeks compared to what they had at baseline, in other words, before they started the drug. And next slide, please. So that table summarizes the results from the COMET study. I put some numbers for the patients who received this Nexviazyme drug and the group that received Myozyme. So the number of patients in each group is similar. Both of these groups of patients have never been treated before. The age range was 83 and up. The Nexviazyme component is Avoglycosidase, and Myozyme is the Avoglycosidase. Very similar. And the table lists numbers as a difference from baseline at week 49 compared to the baseline. So the main outcome measure was the FVC, and that's forced vital capacity, a measure of the lung function in sitting position. The trial also evaluated laying down, but this is not listed here. And you can see that the forced vital capacity for patients who received Nexviazyme was about improved by 2.9%, where the patients who received Myozyme had very small change, less than 1%, like 0.5%. On the 6-minute walk test, the patients on Nexviazyme walked 32 meters more, while patients on Myozyme walked just 2 meters more. The next line in this table is MIP, which is maximum inspiratory pressure, so that's how you inhale. And the number 8.7% in the Nexviazyme group is much larger than the Myozyme group of 4.3. Then MEP is the maximum expiratory pressure. So these numbers, 10.9 and 8.4, kind of look the same. But the next few lines about muscle strength in legs and arms, the numbers for the patients who were on Nexviazyme are much larger than the patients on Lumizyme, like 261 to compare to 154, and then for the arms 174 versus 110. And then the last three rows of this table are questionnaires related to quality of life that patient reported. And the numbers in the two groups are very similar. So although we do see a difference in the numbers of the outcome measures between the patients who were receiving this investigational drug, Nexviazyme, the numbers were larger than the patients who received Lumizyme. None of these reached statistical proof that Nexviazyme is a superior treatment to Lumizyme. However, they're not inferior either. So the FDA did approve that Nexviazyme drug last year. And from what I'm thinking is that in the way that Genzyme Sanofi discontinued Myozyme, they would gradually discontinue Lumizyme as current patients who are currently treated with it will be prescribed Nexviazyme because it has a better outcomes. So we'll see in the next few years, but that's what I'm expecting, that the Lumizyme will be discontinued. Next slide, please. Okay, so the drug Nexviazyme and the active component is, or the engineered enzyme is called Avoglycosidase Alpha. It just got approved by FDA last year. It's also approved by the European Medicine Agency, or EMA. That's the FDA equivalent in Europe. And the drug is approved for patients ages 1 and up, older. And that's late onset Pompe disease. Okay, next slide, please. At the time when Genzyme Sanofi was conducting their Nexviazyme trial, just in the latest years, Amicus was the competitor drug company who conducted clinical trials for Pompe disease. And their drug, well, it has, it was GAA, and during clinical trials, all these investigational drugs have names with letters and numbers, and when eventually they get approved, they get changed to a brand name that's easier to pronounce and remember. So the GAA for the Amicus drug has two components. One is this ATB200, which is the engineered enzyme named glucosidase. And then the other component, the AT2221, is actually a pill that you take together with the infusion, and it serves to stabilize the enzyme, this engineered enzyme, glucosidase alpha. So Amicus applied for FDA approval last year, 2021, and their drug is not approved yet. And I put together a table to compare the last Genzyme Sanofi trial that led to the approval of Nexviazyme versus the Amicus trial results, because how the trial was designed and executed may matter for how it's used and whether it's approved or not. So the Genzyme study involved 100 patients who were treatment naive, means they've never been treated for Pompe disease. And these patients were randomly assigned to receive either the new drug or a Myozyme in a ratio 1:1, so 50% chance. The Amicus trial enrolled or was designed for 123 people, pretty much close to 100 as the Genzyme study, but they were randomized 2:1. So 2/3 of the patients received the Amicus new drug and 1/3 received Myozyme. So the Amicus trial involved both patients who were treated before. So in the table, this is group 2. They had to be at least 2 years on enzyme replace treatment. And it turned out the patients who were enrolled were receiving ERT for, on average, for 7 years. But they also involved the treatment, never treated patients, and that's group 1. So in the Amicus plan to look at the results of both groups together, like combine group 1 and 2. Both the Genzyme and Amicus trials had similar duration, 49 weeks for Genzyme and 52 for Amicus. The primary outcome measure, though, was different in the two trials. So Genzyme was aiming at lung capacity, which showed a difference of 2.4, being better on the new drugs. That will be the Nexviazyme versus Myozyme. And Amicus was looking at the 6-minute walk test and combined for group 1 and 2, so both treated and untreated patients combined. Now, although the two trials had different primary outcome measures, none of them ignored the other measure that came as a secondary. So the 6-minute walk test was secondary measure for Genzyme, and the lung capacity was secondary for Amicus. Now, in the right column for Amicus, I highlighted in green the Amicus results that were significantly different between patients using their drug versus placebo. Well, as I said before, going over the Genzyme study, that none of their outcome measures were supported statistically. Well, the Amicus trial, because their main outcome measures was combining the two groups of patients, like treated and never treated before. The 6-minute walk test for both groups combined had like a 14 meters improvement, but that wasn't statistically significant. But then when they looked at each group separately, it was the group 2 that really improved on their drugs. They walked 17 meters farther. That's the green highlighted. Well, the never treated patients were not different. And then for the lung capacity, the FVC, when group 1 and 2 were combined, patients who received the new drug, the Amicus drug, showed less decline in their breathing function. But if you look at group 1 and 2 separately, it's the group 2 that contributed to this combined group 1 and 2 results. So in other words, the Amicus trial showed that group 2, and these are previously treated patients, showed a big improvement on their drug. But I deliberately put that table with numbers of patients to show you that group 1, and these are never treated patients, was only involved 20 patients, right? And in the Genzyme Sanofi trial, we see that the never treated patients were like 100, and even this number was not enough to reach statistical significance to prove that the drug is better than previous. So with these limitations of the two studies, the Genzyme Sanofi drug, Nexviazyme, got improved, but the Amicus trial did show improvement, especially in the group who was previously treated with Myozyme. So my expectation is that their drug will also be approved. But while we are waiting for that, I want to go over the current experimental treatments. And Michelle, if you can switch to the next slide with gene therapy clinical trials. And I pulled out only the ones that are actively enrolling in the United States. So there are 3 drug companies. One is Scleropius Biopharmaceutical. They have 2 centers in the US. The study is for 8 participants total. Another company is Amicus Therapeutics. They have 3 active centers and 3 more to open. And the whole trial is again for 8 participants. And the third drug company is Spark Therapeutics. And Oregon Health and Science University is one site for this trial, so I'm most familiar with it. And I'm gonna talk about it in my next slide. So Michelle, if you can advance to the next slide, which is the Spark Gene Therapy Clinical Trial. So the investigational drug for the Spark Gene Therapy study is called SPK 3006, and it's AAV viral vector that carries a GAA gene. So what this means, AAV is adeno-associated virus. It's a virus naturally occurring in the environment. So any of us can be exposed to it. However, in humans, it doesn't cause any symptoms, or if any, it will be like cold and flu-like symptoms, so you wouldn't know if you were exposed. This virus is used, it's genetically manipulated to contain healthy GAA genes, and it's used as a way or a method, in this case called the vector, to introduce the healthy genes into the human body. Viruses are normally used for such purposes because viruses can multiply quickly, and they have this other property of infecting cells, or what functionally it translates to is that they insert their genes into the cells. And that's how the healthy GAA gene gets incorporated into the human body cells by infecting them. So in the SPARK study, there are 3 different doses of the study drug for the gene therapy used. The study is designed for 30 participants. Each one gets only 1 dose of the drug. The infusion is once, and it lasts for like 90 minutes. And then the study has a built-in follow-up for about 5 years. So what the gene therapy trials aim at is to, instead of replacing the damaged enzyme, rather introduce healthy genes, and then the body uses these healthy genes to produce the GAA enzyme. So if it happens, then the body will be producing this enzyme, and patients will no more need ERT replacement. And that's the infusion of 4 hours every 2 weeks, expensive, etc. Now the next slide lists the major eligibility criteria for that study. So the patient should be adults, 18 and up, have been receiving Myozyme for at least 2 years. Now that Nexviazyme got just approved, maybe in a couple of years it will change to include people on Nexviazyme for 2 years. It just, because that drug got recently approved, patients don't have 2 years of receiving ERT. And then a couple of major functional criteria are patients to be able to walk between 75 and 500 meters, and their lung capacity to be between 13 and 80% sitting. So these 2 parameters are meant to have patient population who is somewhat affected by the disease, so they don't perform as normal, but still able to do these tests, like, you know, more than walk more than 75 meters, and then FVC to be more than 30, because less than 30, these patients would be more likely on ventilator. So no measurable, more really meaningful lung capacity we can do. And then another criteria is to have this AAV, the virus AAV, antibody negative lab test, because if you have ever been exposed to that virus, naturally, it can happen to anybody, not only Pompe patients, the body immune system would have produced antibodies against this virus. And if we try the gene therapy using the virus, the body will just naturally react and reject the therapy because of the virus. So the next slide just outlines the study design of the SPARK study. So there's one pre-screening visit. It says 1 day, but it means like it's done on 1 day, but it's actually just a blood draw to determine if you have antibodies against the AAV virus. The screening period up to 14 weeks that we evaluate the breathing function and the walking, and we do a baseline assessment of major body systems like heart and liver. We do echocardiogram, liver scans, and then dosing is 1 time. And the follow-up observation about over 5 years. The visits in the beginning are more often and like twice per week, but as time goes by, they're like every 3 months or so. Now, at OHSU, we had 1 patient dosed more than a year ago, and the study involves, or embedded in the study design, is that the patient continues the ERT infusion for about a month after this gene therapy, which we did. And then the ERT was discontinued, and we keep measuring the level of the GAA enzyme over time with this patient, and it's pretty normal. That is to say that this patient is pretty happy not to have any more infusions. And that's about the end of my presentation.
Thank you so much, Diana. Let me switch over to my presentation so we could go through the Q&A. There we are. I see some questions in the chat.
Mm-hmm.
If the audience has any more questions, if you hover over the bottom of the screen, you'll see the Q&A bubble. Click on the bubble and type in your question. I have one question coming in here. It says, what verification has taken place for accuracy of information and message here? Let's see.
Is that about my presentation?
Yeah, I'm not getting any other clarification. Let's see. Let me see if maybe that audience member asked a question here.
Well, I can speak up to everybody. So there's this federal website called clinicaltrials.gov that every clinical trials are required to be registered there. So for the previous Genzyme trials. The results are published, and this website is publicly available, so to me and you, and that's where I pulled out the data from. For the Amicus trial, the results were not published there. I pulled them from the web. The OHSU, we were a site for this trial, but unfortunately our patients, we had one potentially eligible patient and he didn't meet the entry criteria, so we did not enroll that. And unless the results are published in more details, that's all I have. If that was the question, though.
Thanks, Diana. I have another question that has come in. When will the recording be available? The recording will be available in about 3 weeks, hopefully sooner, but if you have registered for this webinar, you will be receiving a link when it is available to watch. Do we have any other questions from the audience? We will give it a second to see if anybody has any additional questions. Here is a question, Diana. Been on Lumizyme since 2006. Do you have any studies showing the different results between Lumizyme and Nexviazyme?
Yeah, this is the COMET study that I presented. So, okay. So in the slide presentation, this is slide 23 that compares people who were on Lumizyme for more than 2 years. Sorry, that study was for patients never been treated. So it's the Amicus trial.
Oh, okay.
But since Amicus hasn't posted results on clinicaltrials.gov, I don't have more detailed numbers for people being from this study, from the Amicus study.
Thank you, Diana. I don't see any more questions coming in. Let me check one more time. Looks like a no. I would like to thank our guest presenter today, Diana. We sincerely appreciate your time, expertise, and everything you do for the neuromuscular community. So thank you very much today. I know we had some technical difficulties, but I'm glad that we were able to work past them and present for the audience today.
Yeah, I'm glad. And if any of you have interest in the current gene therapy study that's going on, you'll be welcome at our site.
We would also like to thank our webinar supporters, Amicus Therapeutics and AskBio. And finally, we would love to hear your comments about this webinar. If you have a smartphone, open your camera and point it at the QR code on the screen. A web page will pop up with a short survey on today's webinar. If you do not have a smartphone, once the webinar is over, a screen will pop up with the survey as well. If anyone has questions after this webinar, please feel free to email them to MDA Engage at mdausa.org, and we will follow back up with you. If you are new to MDA through this program and are diagnosed with a neuromuscular disease under MDA's umbrella, or are a loved one of someone who is diagnosed, we encourage you to stay engaged with MDA. You can do this by visiting mda.org/join and completing a short form. This will keep you up to date on the latest information and education to help keep your family informed in the neuromuscular community. This concludes today's MDA Engage webinar, Updates in Research and Treatments in Pompe Disease. Thanks again, Diana, for your time. Thank you so much to the audience for attending, and have a great rest of your day.
Thank you. You too.
Thank you.